Yes, good morning. Today our team, Group 492, will be presenting our project Crispr AI gene editing tool for off-target activities prediction. Our objective is to develop a model that improves the accuracy of CRISPR or Cas9 editing by predicting unintended DNA cuts, also known as off-target effects. We'll go for the next slide. What is CRISPR? CRISPR is a powerful gene editing technology that uses RNA-guided enzymes like Cas9 to precisely cut DNA at specific locations. Cas9 acts like molecular scissors. sgRNA or single-guide RNA directs Cas9 to right DNA sequence. Target DNA is the exact spot where editing is intended. However, sometimes CRISPR can cut in unintended locations. These are called off-target effects. They can lead to undesired genetic changes. Our project focuses on predicting and minimizing these off-target effects. Aim. The aim of a CRISPR is to accurately predict off-target activities of the sgRNA sequences. By applying deep learning, we aim to design safer and more reliable sgRNAs. This ensures greater precision in CRISPR-based gene editing, overcoming the limitations of older prediction methods. Dataset description. We trained our model on a comprehensive dataset containing sgRNA sequences and a target DNA sequence, and the information whether an off-target will be occurred or not. The data covers different genomic contexts, ensuring the model is robust and generalizable. We applied pre-processing steps such as normalization and sequence encoding to make it suitable for neural networks. The dataset we have used is I2 dataset, which contains an off-target sgRNA DNA sequence, target DNA sequence, and an off-target label, which is which is one for the on-target and zero for the off-target. We'll go for the CRISPR workflow. Here's the overall workflow of the CRISPR. The sequences are encoded, passed through deep learning architecture, and the model predicts whether a given sgRNA will cut the intended site or cause an off-target effect. The pipeline combines multiple AI components to capture both local and long-range sequences patterns. VAT, VAT for CRISPR accuracy, vision transformers. Instead of using only traditional sequences models, we have experimented with vision transformers. Although originally designed for images, VATs can treat DNA or RNA sequences as visual patterns by encoding them into matrices. The self-attention mechanism in VAT allows the model to capture complex spatial relationships within the data, improving prediction accuracy and revealing subtle features missed by other models. The dataset used for the VIT model. We used I2 dataset, which contains sgRNA DNA sequence and a label as well. We used one-hot encoding to represent nucleotides such as A, T, C, G. This encoding transforms the sequences into numerical format compatible with Vision Transformer model. Why are we using VIT instead of CRISPR? The original CRISPR combines CNN BERT. The original CRISPR combines CNN, BERT, GRLAs. Powerful but complex and computationally expensive. For our current milestone, we chose VAT because it offers a simple modular design with fewer moving parts. VAT uses a transformer encoder and a straightforward classification method, making it easier to implement, train, and debug, while still benefiting from attention-based learning. In the future, we plan to integrate to full CRISPR-BERT model for even higher accuracy. We'll go for the model training. We have trained the vision transformer model using our process dataset. The process included data splitting, training with multiple epochs, and optimization using standard deep learning techniques. The model learned to identify patterns in sequence pairs that indicate whether a cut will be an on target or an off target. This was about the model training. On coming to the model output, let me show you a real demo. This was a CRISPR prediction app. This was the home page, and we'll go for the start predicting. And I'll give a perfect match example for the of the two sequences. Look, these two sequences are PAM compatible as the last two sequences are GG, and both of the PAM are PAM is basically the last two sequences of the sgRNA and DNA, and if both of them are matching, the Cas9 cut will happen. Let's go for predicting success. Both of them had same, and yeah, down below we got successfully editing predicted with a confidence score of 95%, and the editing was completed. Now We'll go for the perfect mismatch example. This was mismatch. It was not PAM compatible as it is GA, and both of the PAM were not matching. So basically, it would be Cas9 will not cut here. The Cas9 cannot cut this target sequence. Cas9 cannot cut the input mismatch. And one more thing is the results page. Here you can view the last results, like the last thing we have done. You can view them by uh, confidence score as well as the latest ones, and you can also export them as a CSV format. You can also export them from the CSV format. Here, thank you.